Let me start with the session. And first of all, for the housekeeping, and everybody, when you came in, make sure you're mute yourself so there won't be any noises to interrupt the webinar. A talk, and the talk named as Progress in COVID-19 Therapeutics and Vaccines Research, and with a special review of the hydroxychloroquine and the chloroquine data. So, Dr. Mu, thank you, and go ahead.
Well, thank you, Charles, for your kind introduction. We've given a number of talks over the past few months, and then more recently, we're all interested in therapeutics, drug therapy, and particularly a lot of enthusiasm about hydroxychloroquine and chloroquine. So I would dedicate a large section of today's talk on that topic. Before we Why is my slides not advancing? Before we talk about therapy, let's take a snapshot of what's going on in the COVID-19 world. As of yesterday, the whole world has about 20 million cases of COVID-19 patients, and in the US, about 5.3 or 5.4 million. That makes us 26% of the world COVID-19 patient population. That's pretty significant considering the US population, 4.3% of the world, and even considering GDP. Considering our GDP, which is 15% of the world in 2018, and now 26% of the COVID-19 patients here are here in the US. That's the number of patients. And in terms of deaths, we have 21% of the world COVID-19 deaths in the US, which is still remarkable. But Trump might love to see this chart, because if you look at the middle line percentage-wise over time, it seems that the US has bent the curve and bent it down nicely. It's not so much we've done a good job here. I'm pretty sure we did a good job. But what's making us looking better here is actually what else is going on in the rest of the world. Brazil and India is catching up, and that's what's pushing the percentage curve down for the US. So let's talk about treatment. We need to know this disease is a multi-system disorder. So it's not a simple antiviral therapy alone is going to take care of that. Typically in the first week or so, in the early phase of the disease, it's a viral infection directly resulting from the viral infection. If you do antiviral therapy, you might be hopeful. But then the cytokine storm, the immune response kicks in, and antiviral therapy is not going to do anything anymore. Then you got the endothelial damage, the clotting disorder kicking, the blood pressure problem comes in, and hypotensive. So a multi-system disorder will involve pretty much all the major systems in the body, not just the pulmonary system. Back in April, Lancet published this nice illustration showing a complicated chart of all kinds of medications, including hydroxychloroquine, Remdesivir, corticosteroids, erythromycin, plasma, and stem cells vaccination. You can see chloroquine and hydroxychloroquine is one of the many puzzles being examined. And we're going to review some of the major components here that I put a red circle on. This is an FDA webpage I captured August the 3rd. FDA has a program called Coronavirus Treatment Acceleration Program. So if you are a candidate therapy, FDA will consider you in this program do accelerated consideration for approval. In terms of how early or late stages of clinical studies in the US, there are 180 studies in a late clinical stage, and 40 of them are in early stages. In terms of category of drug types, you have antiviral, about 20 plus studies going on, cell and gene therapy, another 20 plus, immune modulators, about 70 plus. So where does hydroxychloroquine fit into this? Hydroxychloroquine would have direct antiviral effect and as well as immune modulator effect. So it will belong to two categories. And neutralizing antibodies and anticoagulation and other therapy, about 60 plus of studies going on, and there are combination types going on. So a lot of studies are going on, and hydroxychloroquine is only one of the many. So when we evaluate drugs, what do we want? What's the purpose of giving a drug therapy to the patient? First of all, we want to save lives, reduce mortality. That's the hard criteria. If you don't reduce mortality, it makes them feel better, still good. So you can shorten the disease course. If somebody is going to be sick for 2 weeks and you make them sick for only 1 week, that's a progress. So you return them to work sooner. Or you can reduce complications. If someone comes in, you cure them in 1 week, but then they end up with a clotting disorder and stroke and end up paralyzed for life. That's not good either. So you want to reduce complication. And also you want to have minimal drug toxicity. If the treatment is worse than the disease, then this cannot be a good treatment. So that's the purpose of how we look at a drug is good or not. And then the other panel on the right side, I'm showing you in two different colors. The yellow highlighted colors are drugs that have been evaluated by randomized controlled trials. And the bottom white ones, I have not found any randomized controlled trials. Why is randomized controlled trials important? If you recall, back in January and February, there was a story circulated on WeChat, very popular story from China. It talked about at Beijing University, there was a critical care a physician who's their chief, and he got COVID-19 infection, and he gave himself HIV drugs, lopinavir and ritonavir, and he says he got cured. He was better within a few days, and he was convinced that drug helped. What we have to be very careful for infection or any disease that's self-limited. 90% would just get better on their own just by drinking water, being alive. You don't need any treatment, they'll get better. So when you try a drug on your own or small non-randomized trial, it can be fooled very easily to conclude, this drug worked, because this drug might have not worked. You might have just got better on your own. So did this expert, infectious disease or ICU expert, did he turn out to be right? Well, let's look at a clinical trial from China regarding lopinavir and ritonavir. This trial came out very early on, and Dr. Taubin, he's fantastic. I'm a big fan of him, of his. He has published so many landmark studies, and this is one of them. He did a study published in New England Journal of Medicine just a few days or two months after the ICU doctor claims this HIV drug worked. So this was a randomized controlled trial. It happened in January and early February, before the peak happened in China at Jintan Yuan in Wuhan. And they had treated hospitalized patients. So these are not outpatients, they're mild to moderately sick patients. And they were looking at mortality, so very hard criteria. Mortality was similar. There was no improvement in mortality, but let's look at the viral load. The viral load between the treatment group and control group over 28 days, there was no difference. And look at clinical improvements. Did they feel better? Did the fever go away quicker? Did their oxygen requirement improve quicker? No difference, no statistical difference. So now we learned an expert opinion means nothing until it's validated and verified by clinical studies, particularly randomized clinical studies. And this study is widely quoted. And let's look at the next drug, Remdesivir, which was, of course, hyped for a long time. It's going to be the magic drug. I mean, the Xiwan. And then this is a Chinese study that was published in Lancet, which took place in between February the 6th and March 12th, which, as if you remember, the Chinese new case peaked on February the 5th. So they're on the tail end, on the way down, they completed this study. Patients were given the drug within 12 days of symptom onset. Why 12 days? Why waiting that long? Because as you know, even in the United States, as of now, it may take you a week for the test result to come back. And patient might have waited a few days at home. So they were giving that latitude about 12 days to give the treatment. The treatment was for 10 days. And again, looking at the viral load, there was no difference between Remdesivir and the control group. In terms of clinical improvement, nothing. So this was a negative study. So the first randomized double-blinded control study came out from China, multi-center, says Remdesivir was useless. And then the second study was published in the New England Journal of Medicine. This was a multinational study involving the US, Denmark, and many other places, including some Asian countries. They treated for 10-day course, just like the Chinese study. And it was double-blinded, randomized control study, very good quality study. Well, in terms of mortality, same conclusion. There were numerical differences, but no statistical difference. In terms of recovery, the patient who were treated had a median recovery time of 11 days versus 15 days in the control group. So this expensive drug made the patient recover 4 days sooner. And not very impressive, just looking at the numbers. And then the third trial was published a few days later in the New England Journal of Medicine on May the 27th. This time, since the previous study showed some benefit of Remdesivir in clinical improvements, you really cannot give the patient placebo control anymore. So what they did was they did a Remdesivir treatment for 5 days versus 10 days. So they compared these two arms. And in the end, there was no difference in terms of mortality or clinical improvement. So the author's conclusion is we could not make any conclusions because we could not have randomized placebo-controlled study. So we don't know if this drug would have worked better if compared with the placebo. But what we can tell is there was no difference between 5 days and 10 days of treatment. So this is kind of not a very conclusive clinical trials, 3 of them, and one was totally negative. The second one was partially good. The third one is inconclusive. Now let's move on to the next class of drug. That's corticosteroids. Corticosteroids is not an antiviral therapy. It's an immune modulator that reduces the cytokine storm and reduces the inflammation in the body. But corticosteroids did not have a good reputation. The ICU doctors and hospital doctors are very well aware of this. It's been used in ARDS in the past, been used in bacterial sepsis. It's never been conclusive, if not doing any harm. And if you look at some specific viral infection treatment in MERS, it delayed clearance of viral RNA. In SARS, quite a few studies either delays the clearance of the virus or made acute complications, psychosis, diabetes. That's kind of no big deal. It's in a short term, it's reversible. But there's irreversible long-term consequences in some patients. Avascular necrosis of the hip bone, it's a big deal. In our IBD patients, many younger male patients who's been on steroids for long, they're particularly at risk. One SARS series showed 30% of high-dose steroids treated patients had avascular necrosis, and 75% of them had long-term osteoporosis. Influenza trials, corticosteroids made people die faster. Mortality went up, and respiratory syncytial virus in children, there was no clinical benefit. So all this historical data says corticosteroids was not good. And experts being advising not to treat patients unless you really, really have to, COVID-19 patients with steroids. But clearly they were being used in China. And then surprisingly, after all this dismissal, of corticosteroids came this recovery trial from University of Oxford in UK study. They used dexamethasone 6 milligrams a day for 10 days. And for the sick patient who are on a mechanical ventilation, they drop their baseline from 1 down to 0.64. What does that mean? That means there is a 34% reduction of mortality rate. Oh my God. This cheap old drug, it's been long dismissed. It turned out to be better than Remdesivir, which has never been shown to reduce mortality. And then for people who have required oxygen, but not sick enough to be on the mechanical ventilation, it dropped from 1.0 to 0.82. That's 18% mortality drop. And then for the average milder patient was no benefit at all. So that's why the indication for steroids is to give sicker patients who are requiring oxygen supplementation. This is a good study because it's randomized and it's a mortality, very good hard criteria. But they did not have placebo control, I think. And also one critique of this platform, recovery platform, is there are multiple trials going on at the same time. They use the same control group. If something bad, some error was introduced in the control group, the whole series of studies will be biased and errored. So it would be nice to see another group or multi-center study coming out to confirm this steroids finding. So what is dexamethasone? In clinical medicine, we have 3 classes of corticosteroids based on their half-life. You have short-acting, which is cortisone, hydrocortisone, you have to give multiple times a day. You have the intermediate-acting drugs, which most commonly we use prednisone. We're all familiar with that. 0.75 milligrams of dexamethasone is equivalent to 5 milligrams of prednisone. So 6 milligrams of dexamethasone is about 40 milligrams of prednisone. And dexamethasone is the longest half-life, has the longest half-life. We use it for brain swelling, brain tumor, stroke, seizure patients. So that's probably why they chose this dexamethasone instead of the most commonly used prednisone, because it has a longer half-life and probably more steady control of inflammation in the body. Well, let's talk about chloroquine and hydroxychloroquine. Why this minor player among all this treatment, all these drugs, became so prominent and caused so much enthusiasm? Well, there are scientific evidence that this drug has, this class of drug has some antiviral properties and also immune modulation properties that can be hopeful. So there's some scientific basis. Of course, the major reason is we got 2 presidents in the world. One is President Trump in the US, and one is the Brazil president. The Brazil president, you know, he got COVID-19, he got treated, and he said, wow, hydroxychloroquine worked for me, just like the ICU doctor in Beijing who claimed that. Let's review, I'm going to spend quite a bit of time reviewing the hydroxychloroquine literature systematically, so bear with me for that. Before we talk about hydroxychloroquine or chloroquine for treatment of COVID-19 or SARS-CoV-2, let's look at antiviral properties for other viruses. Let's look at the influenza. So there are studies published in Cell Research back in 2013 in mice that shows chloroquine was effective therapeutically, very highly effective therapeutically against influenza, avian influenza, H5N1, but not prophylactically. So let's look at the study, how it's done. On the y-axis, it says wet to dry weight ratio. What does that mean? They are basically weighing the lung tissue. When the lung is inflamed, got pneumonia, it gets wet. So the wet to dry ratio will be high. And when the lungs are normal, the ratio will be normal. Typically, this is not specific for inflammation, but unless you have pulmonary edema from heart failure, you really shouldn't get your lungs wet. So being wet is mostly inflammation. Then they infected the mice with the H5N1 influenza virus. And then the lungs got really wet. So that's the bad one. And then in the next group, they infected with the same virus, but they added chloroquine to it. See, it's not as wet. So they brought it down. Well, they had two other controls. They used the PBS buffer, phosphate buffer, to see if the buffer made the lungs wet. That was not the case. They used another control without viral infection, just used the drug, chloroquine. The lungs were not wet. So it's not drug-induced pulmonary edema. It is infection, and chloroquine did help by reducing the inflammation by almost half. So this is therapeutic, meaning that they gave the drug throughout the course of illness, infection. Prophylactic study, they gave the drug before the infection, but not after. So as you can tell, with or without the chloroquine, the two bar height is the same. The lung got the same degree of wetness. So prophylactically, chloroquine did not prevent influenza, but therapeutically, it did reduce influenza pneumonia. And then let's look at the next study, which came from Singapore, and it's a human clinical trial. It's double-blinded, randomized control trial, very good. But they did not look at the therapeutic effect. They were looking at prevention. They were thinking about, if the influenza is here, this year we got a bad batch of vaccine that does not cover the flu. Why don't we just give chloroquine out and see if it can prevent influenza in the population? I don't want to bore you with all the details. Just look at the P value. All of them were pretty big. So basically, the conclusion was chloroquine did not prevent influenza. It cannot be used as an alternative for vaccination, which is consistent with the mouse study that we just talked about. Now, let's move on to the next virus. We talked about influenza. Let's talk about Zika virus. As you may recall, Zika virus took off some years ago that caused a lot of mosquito-related microcephalus babies in South America. So this is a tissue culture model in vitro study. If you look at Zika infected cells, the cells are infected, about 80% of them will get infected. And then you add increasing amount of chloroquine concentrations from 0 to 50 micrograms. You made some changes, but no statistical difference. But when you reach the concentration of 50 micromolar, then you had a huge drop. The Zika virus infection of cells dropped tremendously. If you look at the viral production, same thing, when you reach 50 micrograms, it dropped. So it looks like chloroquine had some good antiviral properties against influenza and against Zika virus. Then in 2003, now let's move to the next virus, which is SARS. That took place almost 20 years ago when the first SARS took place. There was a hypothesis published in Lancet, infectious disease. This was highly quoted and many studies followed. It says, because the chloroquine has a property of inhibition of cytokines such as tumor necrosis factor alpha and interleukin-6, IL-6, which is now confirmed to be a potent cytokine that causes inflammation. Because chloroquine reduces the cytokine production, it may be a good drug to consider for treatment of SARS and would not have the same osteoporosis, avascular necrosis side effects like steroids. So let's review the data systematically, starting from in vitro studies, cell cultures, and then in vivo studies in animals, and then move on to human studies, and then eventually move on to randomized control human trials. So the first one is a in vitro study using viral E6 cell. You'll see this cell line all the time. It's a green African American African monkey kidney cells. So the white bars are kidney cells that are infected by SARS virus. The black ones were mock infection. There was no infection. So the y-axis is the survival of cells, because the black bar were not infected by virus. They all survived, almost 100% survived. But the white bars dropped, the survival went down. Why did they survive the first one? Because they added 100 micromolar of chloroquine. As the chloroquine dose concentration dropped below 20 micrograms, the cells just started dying dramatically. When it goes down to 4, the cells died, 90% died, and then by the time the drug concentration dropped to 0.8 micrograms, almost 100% cells die. So tissue culture studies, chloroquine was highly effective in controlling SARS-CoV-1 virus. Then the next study is titled Chloroquine is a potent inhibitor of SARS coronavirus and spread, that was published in 2005. So this is before COVID-19. This is about SARS-CoV-1. And this one talk about inhibition of viral infection. When you go up from 0.1 molar micromoles molar concentration to 10, you have increase in potency of viral infection inhibition. And then this is prevention. The drug was given prior to infection. If you give the drug after the infection, which will be equivalent to therapy, then you actually do better. At the same dose, you'll be able to do 50% inhibition instead of 20% inhibition. So again, this drug seems to be highly therapeutic, perhaps less preventative in SARS-CoV-1 infection. The next study came from Wuhan Viral Institute, Wuhan 病毒研究所. It's an in vitro study, and Wang Manli, I see the name, I don't know if this is a male or female, but I see that name quite common in the study. Here, they did a wonderful study, compared chloroquine with the expensive, highly recommended new drug, Remdesivir. And they have 3 different bars. The pink bar is full-time. What does full-time mean? They gave the drug before the viral infection and after. The green bar is entry. They only gave the drug at the time the virus was added to the tissue culture and removed it. And then post-entry, meaning the therapeutic one. The cells did not get the drug before at the time of infection. What's interesting is all 3 categories, chloroquine at the concentration of 10 micromolars were much better than Remdesivir. So you can see from here, if you draw conclusions entirely from in vitro tissue culture studies, not human studies, you can come to conclusions that may not be imaginable. Imagine somebody says chloroquine is better than Remdesivir. Guinea will go crazy. But the data clearly here shows it looks better. But it's not a fair comparison because the molar concentration is different. The same group, Wang Manli from the Wuhan Viral Institute, same kind of in vitro study, they moved on to the next drug, hydroxychloroquine. What is the difference between hydroxychloroquine and chloroquine? Hydroxychloroquine is a derivative, a metabolite of chloroquine, and it's less toxic. So in this study, they look at the toxicity, that they did a dose response curve in terms of cytotoxicity, how much they was gonna kill the monkey kidney cells. They tried the concentration from 3.7 micromoles to up to 400. So somewhere around 33, after that, the toxicity took off. And 50, ED50 is about 200 micromoles or 300. So that gives them idea, wow, our drug was pretty effective at 10 micromoles down here, and the toxicity is high up there. We're never going to go that high dose. That means this drug is really good, has a good therapeutic window in a safety zone. So it was fantastic. And they did an inhibition study of the viral inhibition study, and they showed again ED50 is about 5 or 6 micromolars would be good enough, which is way in a safe zone. So this was a very fascinating study published in a good journal. And but again, it's a basic science study in vitro in tissue culture. We don't know how it's going to translate into human patients. And around the same time, in the Chinese news media, there was a report of 20 cases, clinical study in humans. A short course of chloroquine was highly effective. I could not find the study, so I could only quote this news report. At the time, experts in China suggested, in the absence of any better alternative, the drug is reasonably safe. Why don't we just go ahead and incorporate it into our guidelines, which they did. And so if anybody who wants to see this drug succeed, you know, countries like Brazil, China really wanted to see it, because we don't have any fancy drugs like Remdesivir there. So fast forward to July 2020, in American Journal American Academy of Dermatology, there was a review article summarize the antiviral mechanism of chloroquine and hydroxychloroquine. Why in dermatology? The dermatologists and rheumatologists are very familiar with hydroxychloroquine, Plaquenil. They use it all the time for inflammatory arthritis and lupus, and so they have the experience and knowledge to talk about this drug. So they talked about chloroquine would halt or impede glycosylation of ACE2 receptor. You know, a lot of proteins can't function until it's glycosylated. Sugar is added or carbohydrates added to the protein. And the increased endosomal and lysosomal pH. In the cytosome, inside the cell, the normal pH is neutral, it's about 7.4 or so, but in the lysosome and endosome, where things are getting digested, the pH is usually pretty low, it's about 5 or so. And the chloroquine increases that pH and disables the lysosomal function. And because the virus gets swallowed into the cells, it gets processed by the endosomes. If you mess that mechanism up, the virus may not get out of there. And then there's antigen presenting mechanisms and other immune modulating mechanisms. It was discussed here. So we talked about in vitro studies, we talked about the potential mechanisms, how this drug works. Let's move on to in vivo studies, in animal in vivo studies. This article was published in Nature, very, very sophisticated study from France. They use macaques, a prime, a scary looking monkey here, as the model, because primates are very close to humans. And that's why it was chosen. So they did a comparison. They first did an in vitro study using the same viral cell, the monkey kidney cells, but they also used human airway epithelial cells for comparison. It shows hydroxychloroquine showed good positive antiviral activity in the monkey kidney cells, but not in the human airway epithelium. So there seems to be species difference. What worked in monkeys may not have worked in humans. This is an example. And how about in vivo? In vivo, the drugs were given to the animal, whole animal, not just tissue culture. And they found neither hydroxychloroquine or hydroxychloroquine plus erythromycin reduced the viral load level. So in terms of viral load, it did not show any improvement. And hydroxychloroquine did not protect against acquisition of the infection, meaning it was not good prophylactically either. So as we get into the whole animal study and the human epitheliums, now we're beginning to see negative results. not as encouraging as the initial cell culture studies. And then it came on March the 16th, the famous or infamous French study that shows dramatic improvement of drop of viral load within 5 days, complete viral clearance in 5 days when you use hydroxychloroquine 600 milligrams a day plus erythromycin. And then if you used hydroxychloroquine alone, there's a drop compared to the control group. But if you look at how the study was done, it was only 20 patients, very small sample size. It was not randomized. It was a retrospective observation study. Where is their control? Their control is untreated patients from another center or cases that have refused to join the protocol. So clearly, if you don't have a good quality control group, you really don't have reference to judge how the therapeutic effect happened. So remember this date, March the 16th, and that came out by medRxiv, which is published instantly. And then 3 days later, March the 19th, is when President Trump had the press conference, and this curve was published by JAMA in late May that shows the level of prescription of the number of prescriptions written by physicians. So between March the 16th, the publication of this French study, to the peak of President Trump's press conference, the number of prescriptions just shot up straight line. But very quickly, it did not sustain like that. It dropped down very quickly because many studies is pulling cold water on it. Perhaps it's not so good. This is not enthusiasm of the public. These are prescriptions. At that time, between March the 16th and 19th, the number of prescriptions of hydroxychloroquine and chloroquine shot up in the US dramatically. But right after the press conference and right after FDA approval to write the drug, actually the number of prescriptions went down and regressed to normal level, partly because there was a drug shortage, partly because other studies come out and suggest perhaps this is not as dramatically helpful as people hoped. And then eventually, for safety concerns and lack of benefits or effect, FDA revoked the emergency authorization, emergency use authorization on March the 15th. Now let's look at the next study, which is a positive one. So we look at the French study, which is small, but now let's look at the Henry Ford hospital system in Detroit. They had 2,500 cases, consecutive cases. And here is their result. The green line is hydroxychloroquine alone, and the black line is hydroxychloroquine plus erythromycin. The blue line is erythromycin, the red line is nothing. So it's a control group. It looks like either or the combination of drug did better in terms of patient survival. We're talking about patient deaths. So at the beginning of treatment, everyone was alive. As time goes by, people are dropping off, people are dying. And about 30% of patients died by 28 days. So this obviously study was pouring gasoline on a hotly debated issue, and everyone says it's not good, and how come this study comes out beautifully showing it's working well. But you have to analyze the data more carefully. First of all, 30% hospitalized patient died by the end of 28 days in the control group. That's a little too much. If your control group is skewed, it's so bad, everything else will look better. So there's something wrong with this control group. And if you look at the control group, the red line, most of the people dropped off, died within the first 10 days, which is not typical course. If you look at the Dr. Chaobin study in Lancet, most people died at the end of second week and between second and third week. And so something is not right in this control arm. And that's one. Two, if you think hydroxychloroquine alone or erythromycin alone are effective, when you add the two together, would that line be better? Should be there, not in between. So if you start interrogating this study and its a result, you can't add things up. That's the problem of retrospective study that's not controlled, that's not randomized. So obviously this study does not carry the same weight as the next group of studies I will show you. I'll move on to randomized control studies now. We talked about in vitro study, in vivo study, human studies that are retrospective, not randomized, not controlled. Now we're going to prospective randomized control studies. The first one I could find, I talked about this slide in my previous presentations. It came from Zhejiang 大学学报. Very early on in March. It was a randomized control trial, small sample size, but it's a randomized control trial. They used the hydroxychloroquine 400 milligrams a day, which is very average dose. They did not go crazy with this dose, very appropriate. And their endpoint is the PCR test and see if the viral clearance got any better. They only watched for 7 days, but there was no difference. Actually, in the control group, the group that did not receive hydroxychloroquine, their viral clearance rate was 93%, and the treated group was 86%. So as if this drug be less. Now let's keep moving on to the next randomized control trial. This one came from Brazil, and it was published in JAMA, a good journal. And I gave a lot of weight to Brazilian studies when it comes to hydroxychloroquine or chloroquine, because this drug is used to treat malaria. And Brazil is very, very good studying tropical medicine. As many of us in medicine would know, a disease that's called the Chagas disease. It was discovered about 100 years ago. It was a parasite that causes paralyzed esophagus, very similar to echalation, and also cardiomyopathy. And it's a tropical medicine. Their tropical medicine history goes 100 years. They are very good about that, and I trust their data about hydroxychloroquine or chloroquine. What they did first is compare two dosages of chloroquine, low dose and high dose. If the drug is effective, you think the higher dose would save more lives, right? Well, it turned out just the opposite. Survival, the people who were treated with the higher dose, by end of 2 weeks, 13 days, more than half of them died. And then the group that treated with lower dose, only 20% died. So clearly, high dose is not good. It seems to be killing people. Now, this is another Brazil study. It's published more recently in the New England Journal of Medicine. So they got their study published in very good journals about hydroxychloroquine. It's much bigger size, sample size. It involved 55 hospitals in Brazil. It's a randomized controlled trial. They did hydroxychloroquine plus erythromycin or hydroxychloroquine alone compared to control. There are 3 arms, and they compared the clinical scales in different categories. There's not much difference. So basically, in mild to moderate diseases of COVID-19, either hydroxychloroquine alone or the combination with erythromycin did not improve clinical outcome. So again, we have another negative study, and it's a randomized controlled trial, and it's in humans. Now let's look at this study. It came from China, was published in British Medical Journal on May the 14th. It's hydroxychloroquine with mild to moderate disease. It's a randomized controlled trial. They used very high dose hydroxychloroquine, 1,200 milligrams a day for 3 days, and then 800 milligrams a day for 2 weeks. In mild to moderate diseases, for more severe patients, they treated them for 3 weeks. So if you think the previous drug trials did not use adequate dose, this one is very, very heavy dose. And the first one is patient COVID-19 positive rate. In the beginning, everyone had positive study, PCR study, and with time, the virus cleared. But as we talked about, the virus is going to clear on its own anyway. In most patients, how do you know this drug helped? Well, you compare with the untreated group. There's no difference between the treated arm and untreated arm. And when you look at symptomatic improvement, alleviation of symptoms, there's no difference between the treated group and the control group. So from Brazil to China, the clinical randomized control trial says hydroxychloroquine does not seem to work for COVID-19. And then let's look at the next study. It came from the US, from 40 different states, and from Canada, 3 different provinces. They were giving early, within 4 days of symptom onset, just like Dr. Chai talked about earlier, for viral disease, you gotta treat it early. So typically, 3 to 4 days is pretty good, considering how long it takes for the test result to come back. These are outpatients, so they're milder diseases, not because they're too sick to be saved. And they used hydroxychloroquine, 800 milligrams once, followed by 600 milligrams a day, and then for 4 more days. And again, between the treated group and the placebo group, there was no difference within 2 weeks. So Brazil, 2 studies, China, 1 study, US, 1 study, no difference. Let's look at this one. This one is a Spanish study. It's a multi-center randomized control study. It was done between March and May. Moderate sample size, about 300 patients, non-hospitalized. So these patients are not too sick. They were given the drug within 5 days of symptom onset. And they were giving adequate doses. Again, the treated group and control group, there was no clinical improvement, and there was no clinical improvement of viral load sampled on different days either. So we have data from all over the world, different countries, showing the same data, which obviously is disappointing. We really want this drug to work. And then, if this drug does not work to treat the disease, how about prophylaxis? Just like the hydroxychloroquine study in Singapore, would it work to prevent influenza? Their answer was no. But why don't we do a study about COVID-19? So they selected patients who had known exposure without any adequate protection to a family member, friends, or coworker who had the disease without any protection. Therefore, they are at high risk. The sample size is pretty large. The treatment hydroxychloroquine group is 400 patients and placebo is 400 patients. And as soon as they know they got exposed, they were giving the drugs and were giving placebo and see what happens. Nobody died in either group, so it's hard to say if there's any benefit for survival. And there was 1 patient in the treated group that got hospitalized, 1 patient in an untreated group got hospitalized. Pretty much every category they compared, hydroxychloroquine did not make a difference as a post-exposure prophylactic drug. And this is a randomized controlled trial as well. So we spent a lot of time talking about hydroxychloroquine, and chloroquine clearly from basic science studies in vitro, there was good evidence of antiviral effect, not only to SARS-CoV-2, but to SARS-CoV-1, to influenza, to Zika virus. But when you move on to human studies and randomized control trials, not a single randomized control trial showed benefit of the drug. But I would say we should not dismiss the drug completely. We only know it probably does not work by itself. What about the combination? I'll give you an example. Here's a combination study from Hong Kong. It was just published in May in Lancet. They used the HIV drug, which we already know does not work, and they used ribavirin. For those of us who practice hepatology and gastroenterology, we're very familiar with this drug. This is an antiviral, broad spectrum antiviral we use to treat hepatitis C as an adjuvant drug. And interferon we use for hepatitis C treatment and hepatitis B for many years as well. So they put the three antiviral drugs together. The control was not a placebo. The control is the HIV drug, which we know it doesn't work. So they use that as a control, fantastic control. So here we go. The viral load in the control group, untreated group, was this high. It drops down slowly. In the combination treated group, it dropped down much more rapidly. And in terms of symptomatic score, they use this national early warning score. I never knew about this, but I read about it. It's using respiratory and CO2 retention and a few other parameters to calculate its score. At baseline, both the treatment group and control group had the same score, but over time it dropped down to 0 quickly. In the treated group, it dropped down to 0 on an average in 4 days. In the untreated group, it went down by 8 days. It looks like they shortened the clinical course by half, which is fantastic. So the lesson from here is that a drug may not work individually, but may work as a combination. It's like a puzzle. There are so many drugs out there, and somebody is going to find a combination that might find utility for hydroxychloroquine or chloroquine. Until then, we'll know for sure if we should dismiss that drug completely. But for the time being, it's probably not wise to give that drug as a monotherapy. All right, we're down with hydroxychloroquine. Let's talk about the next group of treatment, anticoagulation. The anticoagulation, we don't have randomized controlled trials, but clinical observations by clinicians, by previous studies show there are a lot of people with COVID-19 infection form clots in the body, which is dangerous and kills patients. The first clinical observation came from Dr. Taubin's report in early March. His team find D-dimer as an indicator of people who's going to either survive or who's going to die. They find if your D-dimer level keeps going up like that, you're more likely to die. If your D-dimer level stays flat, very low, you're most likely going to survive. What is D-dimer? D-dimer is the degradation product of clots. And so this study shows some clotting events is happening in these patients, and it plays a big role in causing patient deaths. Well, soon after that, the German study, which was widely quoted and talked about by the news media, and they examined 12 patients who died, consecutive patients. They did not pick and choose. All 12 patients died one after another. They did autopsy on them. Over half of them had DVT. What is DVT? Deep venous thrombosis, clots in the legs. What would this do? If this clot falls off and goes into the lungs, it causes PE, pulmonary emboli, and it will kill the patients. So in their autopsy, they found that at least 30% of patients had massive PE, and they determined the PE was their direct cause of death. Here are some of the pictures they showed. Here's PE, saddle PE in major pulmonary arteries. Here's the in the veins in the lower extremity, big clots. And then if you have clots in the small blood vessels, the patient will have purple blue toes. And in GI, I wanted to show you a GI case in the world of gastroenterology and hepatology. You can have portal vein thrombosis. The liver has two sources of blood supplies. 1/3 come from the hepatic artery, 2/3 come from the portal vein. So when you plug up the portal vein, that's a big blow to the liver. And if you got viral hepatitis on top of that, that makes the liver very unhappy. So in terms of anticoagulation and the clotting, how would they help? This Dutch study published in May shows in the ICU, after 20 days in the ICU, about 60% of them will have some kind of clotting disorder, DVT or PE, despite the routine thrombosis prophylaxis, because in our ICU patient, a hospitalized patient, they get routine clotting preventing treatment by heparin shots. And for ward patients, not as sick, they stay on the floor, not intubated, they get about 10% clotting complications despite prophylaxis. So clearly, this is a big problem. And unfortunately, we don't have a randomized controlled trial to show anticoagulation improves survival, but we do have retrospective observation studies. This is from Mount Sinai in New York. For mechanically ventilated patient, if you give them treatment dose, meaning not the prophylactic dose, then you can improve the survival, the drop off, the patient don't die as fast. And for people who only received prophylactic dose, they die off much quicker and faster. And it seems to be most effective in the sicker patients, which is consistent with the previous Dutch study that shows clotting complications happen more often in the sicker patients. The title here is association. They put the two together. When you treat the patient with treatment dose, you have better chance to survive. Because it's not a randomized control study, they cannot determine that's the causal and result effect. That's why they call this association. Now, let's talk about the next class of drug or treatment. That's convalescent plasma. As we learned from the Ebola years, if a patient survived and you use that patient's blood plasma, which contains antibodies against the pathogen, and you can save the next patient. So this was talked about quite a bit. Is this approach really effective? Does this approach save lives? We don't know until we do clinical studies. So here we do have some studies. This one is match control study. It's not a randomized control study. It's observation. It's from Mount Sinai again. It shows if the patient is very sick, intubated, actually it helps. It drops from the rate of deaths at 1.0 down to minus 1.66. So for the, sorry, I got it confused. And then for the intubated patient, it did not make much difference. So for people who are not very sick, this approach seems to have helped. But for the very ill, very sick patient, plasma did not help at all. And their observation is confirmed by this randomized study. You'll be amazed as you review the literature. Our Chinese physician from China has done tremendous amount of work. Pretty much anything, everything you can think about, there was a study, and oftentimes it's from China. They're a good randomized control study. This finds this randomized control study. It shows if you're having life-threatening disease, the plasma treatment did not make any difference. So very much identical to the observation on Mount Sinai in New York. If you're very ill, but not critically ill, the plasma helps. So the plasma make you feel better, make you recover quicker, but it doesn't seem to be able to save you. If you're sick enough, you're gonna die, you're gonna die. So that's my take based on these two studies. The next study was from Netherlands about plasma, convalescent plasma. It's a randomized control study, well designed, but after they enrolled 86 patients, they stopped the trial completely, they halted it. Why? Because they realized based on their inclusion criteria, you don't give the plasma to any mild patient who walks through the door. You wait until they get sicker, which usually happens around 10 days. But by then, 79% of the patients already have their own antibody of a similar titer to the plasma donors. I say, what's the point of treating them if they are already producing this much protective antibodies already? So they stopped the trial. We never know the results. but their conclusion is probably not going to make a difference. And so we talked about not a lot of drugs, and then there's this page, this slide shows all the drugs that are patented in China in clinical trials, and these all holds Chinese patent. As I go through them, some of them syrup and probably a lot of herbal medicine. They're not the new synthetic drug. And this left panel shows the number of clinical trials been going up rapidly. So a lot of studies going on in China. Let's talk about immunity and vaccinations. There was a lot of concern about how durable the antibody would be. If you develop the antibody, you lose it, it's not going to be good. Just like the flu vaccination, you got to do it every year because it's not going to protect you for long. Well, this study published in Nature gave us some encouraging findings here. They found a patient, one individual, not too many, who was infected by SARS-CoV-1, the first SARS, back in 2003. They were able to isolate the memory B cells from this individual, and they were able to produce a lot of different monoclonal antibodies against SARS-CoV-2, even though the infection was SARS-CoV-1, so there was cross-reactivity. And one of the monoclonal antibody, they called it S309, is highly effective. And they determined the epitope of this antibody was spike protein receptor binding domain. So this study is very, very significant. One, it shows the B cell may have very lasting, long-lasting memory to fight against the virus years later. Two, there's cross-reactivity between SARS-CoV-1 and SARS-CoV-2. Three, they know which antigen epitope to go after. That gives us guidance how to develop vaccines. In my previous talks, I gave the example of hepatitis B. There are different antibodies against hepatitis B. There's surface antibody, there's E antibody, and there's core antibody. If you go after the run target, you go after the core antibody or E antibody, they're useless. They don't protect you. You need to be going after the surface antigen to produce surface antibody. So based on this earlier studies, the scientists know they need to go after the spike protein to produce spike antibody, particularly the receptor binding domain. So this saves a lot of time for antibody generations. And so to develop vaccination, there are different strategies. There's time-tested, inactivated viral vaccines. The good examples are hepatitis A antibody and the polio vaccines, the Salk vaccine. But it's time-tested, it's effective. The second approach is recombinant. Instead of using the whole virus, which sometimes can be dangerous, you make DNA construct to produce the antigen proteins, purified antigen protein, and you'll be able to generate antibody against the virus. It will be effective. The classic example is hepatitis B virus. And I think the Cubans are very good about producing the recombinant DNA that produces surface antigen that made effective hepatitis B vaccinations. Now we're in a new era. This is nucleic acid vaccinations. Instead of giving protein, the antigen to cause antibody production, now the new approach is to give nucleic acid, either DNA or mRNA. The advantage obviously is tremendous, because if you were to make recombinant DNA, you have to make the DNA construct, and you have to do in vitro translation to make the protein at industrial scale, which will cost time, money. And if you deliver the DNA or mRNA into the patients for vaccination, you're setting blueprints to the patient. You tell them, here is how you make the protein by using your own cells. I'm not going to make it for you. It saves a lot of cost and manufacturing process. And there are many other advantages we'll talk about, but there's a huge difference here. But so far, it has never been a vaccine that's proved yet in the world that's DNA-based or mRNA-based yet. But because of these new construct, vaccines can be developed much quicker. Here is a WHO report I put off the internet. There's a website here. There are over about 160 vaccines in development, and 30 of them are in clinical trials. And I'm going to talk about a few. The University of Oxford virus, which is a viral vector delivery, and the Moderna vaccine, and then there's the CanSino, the Chinese vaccination vaccine that was published, also uses adenovirus, type 5 adenovirus as a vector. The first vaccines came from China. That's they monitored the antibody production. They use high dose vaccination, middle dose, and high doses. They detect the antibody, the ELISA, against the receptor. And the produce the patients, not 94. And same thing with low dose. So why is there a difference between the antibody when you measure it by purified antigen or measure by live virus? If you look at this study, that will give you the answer. This study was published in Proceedings of the National Academy of Sciences in May. It says, under normal conditions, the viral protein is folded. The receptive binding domain is lying down, is hidden to evade immune system. Only when it's getting ready to be bound with the ACE2 inhibitor, to be exposed. So when you add the antibody to live virus that's not bound on the receptor binding domain is hidden. So it's not always effective. It's tricky to make antibody, and not all the epitopes will be exposed based on the protein folding. This is a phase 2 study of the same Chinese vaccine. It was just published in Lancet in late July, measured by the geometric mean antibody titer, placebo, no antibody, and different doses produce high using different assays. So the vaccination not only produced humoral antibody response, but also produced T-cell responses. Seems to be a good antibody, and Dr. Fauci was talking about if you can produce a vaccine that will protect 50% of the patients, 50%, even at the low dose. So this vaccine seems to be a good candidate. Now, the second vaccine was from University of Oxford, and it used the same adenovirus as a vector to deliver, and they produced very good viral titer, very comparable to convalescent plasma from patients. So this vaccine seems to be very hopeful too. This is a combined published. Now let's move on to the next one, the final one of the nucleic acid vaccination. It's from Moderna. It's published in New England Journal of Medicine. And they use different doses, 25, 100, and 250. They all seem to have produced nice titers of antibody. The last call is convalescent antibody. vaccines. It gives you the blueprint to produce your Moderna mRNA trial. This is the newest type of the only nucleic acid vaccinations being ever published. It delivers mRNA that encodes the spike protein, so the host cells will produce the antigen and the vaccination for themselves. They can use a lower dose, not milligrams, usually it's micrograms. Even at the lowest dose, it produces very high antibody titers and also provides a stronger T-cell response. This one just came off the press only 2 days ago in JAMA, so I added it today. It's a Chinese study using inactivated virus. Seems to be very helpful. So now among all the randomized controlled trials published for vaccination, there are 2 of them came from China, 1 came from UK, and 1 from the US. There was much hype about the Russian vaccinations, but I could not find any randomized or any publications. Instead of doing my own summary, I'm going to use the NIH guidelines. The NIH guidelines published at this website. If you're interested, you can go there to summarize what we talked about in our treatment protocol. Dexamethasone is recommended for people who require oxygen, so mildly to moderately sick patients, all the way to very sick patient in mechanical ventilation. The dosage is 6 milligrams every day for 10 days. And NIH guidelines now say you can use other steroids too, prednisone, methylprednisolone, or hydrocortisone. It doesn't have to be dexamethasone. Remdesivir is in short supply, so it's not going to be given to the very mildly ill patients. It's going to be given to someone at least on some oxygen supply. But NIH says for very sick patients who are on high oxygen flow or mechanical ventilations, the data is not clear if they save lives. Like we talked about, it makes you feel better, but doesn't really save lives for the very sick people who may not make it. And NIH says the physicians can make their own judgment whether or not to give the drug to the patient, because it's not going to do much good, probably. And now it's recommended for 5 days. Remember the last article from New England Journal of Medicine that compared 5 days to 10 days? There were no difference between the 2 groups. So now it's recommendations for 5 days instead of 10 days. Anticoagulation thrombotic treatment. There was no randomized controlled trials, so the evidence level is low. So NIH does not seem to have made any specific, more aggressive treatment. And there was no guidelines about if your D-dimer is high, you anticoagulate them. It was pretty vague. Clinical indications used for management of these people, and they do not recommend anticoagulation for outpatients, and they do not recommend routine DVT prophylaxis when discharging a patient. In terms of hydroxychloroquine and chloroquine, the recommendation says they recommend against the use of these drugs outside clinical trials. Do a clinical study first. And they do not recommend the use of the combination of erythromycin and hydroxychloroquine either. And they highly recommend against the use of high-dose chloroquine, 600 milligrams BID. That's where the Brazil study came in, because higher dose actually killed more patients. And the other drugs are also recommended not to use outside clinical trials due to insufficient data, including the HIV drug, stem cell treatment, kinase inhibitors, plasma, immunoglobulins, IL-1, IL-6 inhibitors, and other adjuvant therapies, vitamins, zinc. And so the clinical data is not sufficient to make a recommendation for or against. Well, thank you. That was a, sorry about the interruption. I don't know what happened. I got kicked out twice. That's my
James.